All right, well, it's a great pleasure to introduce Bob Datta from very close by, our neighbor, and actually my neighbor and my friend. So Bob got his PhD and his MD from Harvard Medical School in Michael Greenberg's lab. He went on to a postdoc with Richard Axel. He subsequently came back to Harvard, and is an associate professor at Harvard Medical School. And Bob has done amazing work in many, many different domains, so ranging from understanding olfactory codes to characterizing quantitative behavior to basically looking across from the sensory to the behavioral arc overall and everything in between. And he's going to tell us, I think, about some of the stuff in between in dopamine systems now. So welcome, Bob. And before, Bob, I let you go on with your talk, I think Lisa is here, so I just want to acknowledge her being present. So hi, Lisa.
Excellent. Can everyone hear me? We're good? Excellent. So thank you, Ela, for that kind introduction and for the invitation to come and speak. And congratulations both to you and to everyone here on the founding of this ICoN Center. I think it's the right moment to think about a sort of integrative computational neuroscience. I will say that I was really excited by your first slide where you kind of described trying to integrate data across many levels, going all the way down to genes and molecules. I think that's an idea that many of us have talked about for a very, very long time and haven't really reduced to practice. And it feels like we're having a moment in neuroscience where that's really possible. So I'm excited for this new ICoN Center, and I look forward to seeing the great work that comes out of it. OK, so let's get going. I have to stand this side? I think I do. Oh, there we go. Excellent. OK, so the behavior of animals in the natural world, where they're free to do what they want, when they want, is obviously remarkably complex. And it's because of this complexity that to us as humans, it often appears that their behavior is fundamentally inscrutable. But of course, the ethologists working 50, 60 years ago hypothesized that spontaneous naturalistic behavior like this actually has a ton of underlying structure. They believed that spontaneous behavior is actually composed out of a series of behavioral modules, brief, three-dimensional, stereotyped, repeated units or motifs of action or movement that are concatenated one after the other in time to build complex behavioral sequences, and which are organized hierarchically in time, thereby affording behavior a kind of spatiotemporal organization that's selected for by evolution and allows animals both to kind of deal with the challenges of their particular ecological niche, but also enables them just enough flexibility to adapt to changes in context and contingency. They also made an argument that I think all of us will find very intuitive, which is that if indeed there is a kind of underlying structure to behavior, it must somehow arise in the nervous system. But despite the fact that this sentence was written now nearly 60 years ago, we still, I would argue, have only the most superficial understanding of how the brain composes spontaneous behavior. And here by spontaneous behavior, I mean the kind of behavior that you and I and almost every creature spends most of our day doing. The kind of behavior that happens in the absence of a task, without obvious and explicit rewards, sometimes without obvious goals. The kind of behavior that happens when you're not confined behaviorally or psychologically or physically. This question, how the brain composes naturalistic behavior has really haunted my lab for the last decade. And so to begin to address this question, we've been building tools that allow us to characterize the underlying modular basis for mouse behavior in the laboratory. And the idea is that if we can begin to do that, we can begin to test this kind of ethological hypothesis that behavior is built out of parts. The tool we've built and we've been working on is called motion sequencing, or MoSeq for short. We've published a bunch on this technique, and so I'm not going to describe it to you in any great detail at all, because I want to show you just the data. But at a very high level, MoSeq is an unsupervised machine learning algorithm that, based on the spatiotemporal structure of behavioral data alone, without any human supervision or intervention, is capable of discovering an optimal set of behavioral modules, or what we call behavioral syllables, out of which behavior is composed in any given experiment. The behavioral data we tend to feed motion sequencing is 3D imaging of mice as they explore simple arenas like this circular open field over here. And we obtain these images, these 3D images, by placing depth cameras, which we buy from Best Buy for like $100, over buckets that we get for $5 from Home Depot. And you can see the output of one of these depth cameras here. Here, heat mapped over each pixel of the mouse is its height at any given position. And you can very much get the sense that these cameras, even though they're imperfect in many ways, give you a rich representation of the animal's ongoing three-dimensional pose dynamics. And again, we feed these 3D data to the MoSeq algorithm, which is a generative time series model for behavior that instantiates this ethological hypothesis that behavior is composed out of modules that tumble one after the other somewhat predictably in time. And by fitting this model through unsupervised techniques, MoSeq discovers this kind of parts list for behavior. In a typical open field experiment, it discovers maybe 40 or 50 of these behavioral syllables, which correspond to human identifiable behaviors, things like pauses, dives, and runs. The average duration of each one of these little behavioral syllables is 300, 400, 500 milliseconds. And so MoSeq teaches us about a kind of sub-second architecture that seems to be characteristic of mouse behavior. And we can synthesize for any given experiment the entire output of motion sequencing into one of these behavioral state maps. Here, each node is a behavioral syllable. The diameter of each one of these syllables represents how often within an experiment that particular syllable is used. And the thickness of these directional yellow arrows indicates the observed likelihood of transition over time from one behavioral syllable to the next during the experiment. And so I hope you can tell just by inspection that the behavior of mice in the laboratory is captured by MoSeq, just like the behavior of mice out in the real world is characterized by having an enormous amount of structure. And the question we want to answer is, where does the structure come from? How does the brain build this? And that has got us thinking about parts of the brain that are involved in building behavior, in specific corticobasal ganglia loops, and in particular the input nucleus to the basal ganglia. That's the part of the brain here in blue called the striatum, because of its well-known roles both in action selection and in motor control, which of course are going to be functions that are critical to composing spontaneous naturalistic behavior. We've focused on a specific subregion of the striatum, a part of the striatum called the dorsolateral striatum. That's for a couple of reasons. First, dorsolateral striatum, or DLS, gets inputs from proprioceptive and motor cortices, suggesting the dorsolateral striatum has access to the kind of information you'd want if you wanted to organize your 3D pose dynamics. But also because work by others, in particular Kent Berridge, has suggested that during simple spontaneous behaviors like grooming, the dorsolateral striatum might play an important role kind of in organizing those behaviors. And so for all those reasons, we wanted to test the hypothesis that maybe the DLS contains within it neural correlates for behavioral syllables, behavioral grammar, right, the sequence in which syllables are expressed, or both. So to address this question, we have rendered, together with our collaborators, motion sequencing compatible with all forms of neural recording. And so now we can use kind of MoSeq as a general purpose platform for querying brain behavior relationships during spontaneous behaviors. So let me just show you an example of us doing this that's shown here. So here on the upper left is going to be a mouse running around in one of these buckets. This is obviously a 3D image. Here we're going to extract the 3D movie of the mouse. We're going to align it so its nose is to the right and its tail is to the left, and we're going to center it in this image. You can tell that this mouse is wearing a hat out of which is coming a cable. And that's because we've implanted a miniscope within its dorsolateral striatum, and you can see what the miniscope is visualizing here. Here you're looking at calcium transients and individual spiny projection neurons in dorsolateral striatum. And here in the center, we can line these two data streams up. Here on the bottom, looking at calcium transients and individual spiny projection neurons in DLS. And up above is our post hoc identification of each behavioral syllable as it's expressed. Sorry, I'm a little bit under the weather, so I just want to, perfect. Okay, and so what you can see, even just by inspection, is that there seems to be some interesting relationships between population level activity in these neurons and the behavior that animals express as they freely behave. It's as if, as animals switch from one behavioral syllable to the next, there's a switching in the ensembles of neurons that are activated such that a new ensemble of neurons is kind of dialed in as the animal switches from one behavioral syllable to the next. And if we formally analyze the data, that's exactly what seems to be true. And indeed, if we look at the ensembles themselves, it turns out we can use the identity of each syllable-specific neural ensemble to make accurate predictions about the specific 3D behavior the animal is expressing at any given moment in time. In other words, we can kind of look down at the animal's brain, and I can tell you which syllable it's expressing on a moment-by-moment basis. In addition, this neural data contains within it information about the specific sequence context in which each syllable is expressed, And if we lesion the dorsolateral striatum, animals continue to appropriately express behavioral syllables from a kinematic perspective, but their ordering is totally incorrect, preventing them from generating kind of adaptive sensory-driven behaviors. And so what this tells us is that indeed the dorsolateral striatum seems like it's an important place for organizing spontaneous behavioral sequences, but it fails to tell us some of the answers to some of the key kind of remaining questions about where this structure comes from. For example, why are some behavioral syllables used more than others? Why do some behavioral syllables like this one tend to participate in highly deterministic, very predictable behavioral sequences, going from here to here to here, whereas other behavioral syllables tend to be connected to a bunch of other behavioral syllables more probabilistically or randomly, seemingly participating in behavioral sequences kind of more willy-nilly. Where does all that structure come from? And so thinking about that problem got us thinking about the major neuromodulator that influences the striatum, and that is, of course, dopamine, which is well known to influence both population-level activity in medium spiny neurons in the striatum, and to be capable of inducing plasticity in those very same neurons, which is why dopamine has been implicated in a large number of different contexts as a teaching signal. The dopamine that talks to dorsolateral striatum largely arises from neurons that live in the midbrain in substantia nigra pars compacta, as shown here. And there's reasonable reason to believe that dopamine might play an important role in organizing spontaneous behavior. And we know that from thinking just about what the behaviors that are changed in patients with Parkinson's disease, right? Those patients have difficulties in initiating new movements, in invigorating their movements, and in appropriately sequencing their movements. That being said, we actually know very little about the role of dopamine during spontaneous behavior in animal models. There's basically two lines of thought about what dopamine might be doing in animal models. The first comes from work of folks like Rui Costa and Dan Dombeck, who've observed that as animals are stopped and then generate a new movement, there tends to be a little flicker in dopamine, suggesting that maybe these dopamine flickers that are associated with movement initiation are causal. And indeed, if you kind of optogenetically induce a flood of dopamine in the striatum, animals will begin a new movement. There's another kind of non-mutually exclusive set of ideas coming from Henry Yin's lab that maybe dopamine dynamics are actually quite continuous, and the continuous variation in dopamine actually specifies the animal's ongoing movement kinematics. But again, as I've mentioned, there actually hasn't been that many careful kind of recordings of dopamine as animals are just spontaneously behaving in any context. So the degree to which either of these is kind of a general rule remains a little bit of a mystery. What is true and what we know a lot about from 40 years of work is something that I've already mentioned, which is that dopamine plays this key role as a teaching signal and is capable of shaping animals' future behaviors in order to assist animals in kind of maximizing their ability to obtain rewards. But it's hard to think about the kind of role dopamine might play as a teaching signal, helping animals to obtain reward, in a context like the open field that I showed you, where an animal is running around in the dark with no task, no goals, and certainly no reward, right? And so most of our attention has been focused on trying to understand whether or not dopamine during spontaneous behavior relates to initiation or kinematics. Okay, so we began thinking about this, and we decided to do a very simple experiment, which is just to repeat the experiment I showed you before, but now to record dopamine transients, specifically in dorsolateral striatum, through the use of the dopamine reporter D-Light. And so here, we're doing fiber photometry. You can see the mouse running around in the bucket over here. And here, you're looking at dopamine transients as reported by D-Light, locked to syllables which are being identified post hoc. And what you can see just by inspection, right, is that there are far too many dopamine transients to be explained by movement initiation alone. In addition, if we do a ton of math, which we did, we have a very difficult time identifying any kinematic parameters that seem to be encoded by these dopamine fluctuations, with one exception, and that exception is velocity. So what's interesting is that if we bin the dopamine transients at relatively short timescales, at timescales of 5 to 10 seconds, we actually see a negative correlation between the amplitude or number of dopamine transients the animal's experiencing and its speed. In other words, if there's a burst of dopamine during spontaneous behavior, animals actually tend to slow down. And that kind of weird counterintuitive result is something that had been observed by others in the field before. But what's interesting is that if we bin the data over longer and longer timescales, this relationship actually seems to invert. And now we have the kind of comfortable positive correlation between dopamine and movement that all of us were taught kind of in our neuroscience classes, where dopamine is thought to kind of broadly invigorate or enable in some way the animal's ability to generate movement. Okay, what about movement initiation per se? So this is what we see if we look at movement initiations. The x-axis here is time. Time 0 is the moment at which the animal generates a new movement after being still. And indeed, we can see a nice little dopamine flicker, as has been observed in the past, associated with the initiation of a brand new movement. But the amplitude of this flicker is much smaller than the amplitudes we observe if we actually tee off this data on all behavioral transitions. So here, time 0 is actually the transition from any syllable into any other syllable, right? So the animal's moving here and it's moving here, just that the movements are different. And what you can see are these nice large amplitude changes in dopamine that systematically reflect when animals switch from expressing one behavioral syllable to the next. It's as if the dopamine kind of exhausts itself as the animal ends one syllable, and then there's this large upsurge in dopamine as animals enter into and begin to express the very next syllable. So this is what we see on average. We see this large systematic fluctuation in dopamine across all syllable transitions. This isn't actually what we see if we look at individual syllables. If we look at individual syllables, we see a lot of variation in the dopamine amplitudes that are on average associated with any given syllable. So here we're identifying 40 different behavioral syllables in this experiment. And if we just look at this very top behavioral syllable, which turns out to be a rear, when animals begin to express this rear, dopamine kind of quickly ramps up to this very high amplitude, and then characteristically falls. In contrast, if you look at this syllable down here at the bottom, this turns out to be a pause. When animals express a pause, their dopamine tends to be pretty low, then slowly during the expression of the pause, it ramps up. And if we look at any one of these syllables, we'll see associated with it a characteristic dopamine waveform. Now, this might give you the idea that we should be able to use these waveforms or their amplitudes to predict which behavioral syllable the animal is expressing at any moment in time. In much the same way as I told you that if we look at actual neural activity in spiny projection neurons, I can make accurate predictions about which 3D behavior animals are expressing. So if we try to play that same trick with these dopamine transients, it turns out we can't make predictions. I can't tell you which behavior the animal is expressing on a moment-by-moment basis based upon the dopamine waveform that's associated with that behavior. And the reason why becomes obvious if we just look at different instances of this particular behavioral syllable. What we find is that there's a huge amount of variability in terms of the amplitude and waveform in dopamine associated with any given behavior. Yes, indeed, on some instances of this rear, you see this kind of high amplitude dopamine transient, but on many instances, you actually see dopamine going in the opposite direction. And that holds true for all of the behavioral syllables. And so we can't make predictions on an instance-by-instance basis of which syllable is being expressed based upon the dopamine waveform. And this is broadly consistent with my previous claim that it does not appear that dopamine is encoding the kinematics of the animal's movement. Here are 4 separate examples of the animal generating the same rear up behavior. And yet, in all 4 of these cases, there's a different dopamine waveform that's associated with it, right? So these dopamine waveforms can't be encoding kinematics. So now we're in this kind of weird situation, right? It's clear there's a strong temporal correlation between dopamine kind of flickering as animals switch from one syllable to the next during spontaneous behavior. But it's unclear what these flickers mean to the animal. They certainly don't encode kinematics, and they don't encode the ongoing behavioral syllable. So in order to kind of clarify our own thinking, we did a simple calibration experiment. So all we did was we took some chocolate chips, and we just threw them into the bucket. And then we measured dopamine transients as animals consumed these delicious and surprising and rewarding treats. And what we observed is that the dopamine fluctuations that were elicited when the animals consumed these surprising rewards, which clearly in a different context are capable of shaping the animal's behavior, were of roughly the same amplitude as the fluctuations that we were observing in dorsolateral striatum during spontaneous behavior. And that suggested to us a really surprising possibility. Maybe these dopamine fluctuations aren't actually controlling the animal's movements in the moment. Maybe they're instead controlling the animal's movements in the future, right, by reinforcing in some way the syllables with which they are associated on any given syllable instance. If that is true, he would make a very simple and basic prediction, which is that those syllables that are associated with the most dopamine on average during the experiment are precisely those syllables that should be used the most on average during the experiment. And in fact, as you can see here on the left, that's true. The x-axis here is dopamine. The y-axis here is how often a syllable is used. Each dot here is a syllable mouse pair. And indeed, we see this nice positive correlation revealing that those syllables that are used the most are those that on average are associated with the most dopamine. And what's super interesting is that this relationship between dopamine and syllable usage holds true on a moment-to-moment basis. So if the animal is just kind of generating behavior, and on a particular instance of a syllable, say that rear I showed you, there's a high amplitude dopamine transient, then that particular rear is going to be used more for about the next 3 minutes, as shown here by this orange line. And if that very same rear on some different instance is associated with a relatively low dopamine transient, then over the next 3 minutes, that specific syllable will be used less. Now, dopamine, of course, is not just important to reinforcement. It's important to action selection in a whole variety of different contexts that has been shown. And so we wondered whether or not these dopamine transients that were associated with individual syllables played any role in determining the types of sequences that syllables were embedded in. And so we just asked, when, on average, were those syllables that were associated with high levels of dopamine, do they tend to be embedded in more predictable sequences or more random sequences? And it turns out, as you can see here, that there's a nice positive correlation between how much dopamine on average is associated with a syllable and the variability of the behavioral choices that animals would make in the future, such that syllables associated with high dopamine tend to be embedded in more variable behavioral sequences, and syllables that are associated with low dopamine tend to be expressed as part of more predictable, less variable, less entropic sequences. And again, this effect seems to hold true on a moment-by-moment basis. If you're expressing that rearing syllable I was mentioning to you before, and you happen to be associated with a high amplitude dopamine peak, the next 2, 3, or 4 behavioral choices you make tend to be more variable ones. You tend to make a kind of more unusual next behavioral choice, as shown here by this orange line. And conversely, if that same rear is associated with relatively low levels of dopamine, the next thing you do tends to be more predictable and kind of more ho-hum. So now I've described to you 3 things that appear to be correlated with dopamine. Those 3 things are the animal's velocity, the usage of individual syllables on timescales of minutes, and behavioral variability in terms of sequence variability evolving on timescales of seconds. And we've built encoding and decoding models that kind of validate all the correlations that I've just described. I'm not going to show you any of those models. Instead, I want to turn to our attempt to do a causal experiment. What we wanted to do was to somehow manipulate dopamine specifically during the expression of a given syllable to ask whether that manipulation would affect velocity, usage, or variability. So in order to do this, we've taken advantage of the magic of deep learning, which allows us now to render motion sequencing closed loop. So now we can look down at animals freely behaving and just running around in a bucket, and I can tell you with about a 50 millisecond latency or so which specific behavioral syllable the animal is expressing at any moment in time. And based upon which syllable it's expressing, I can pick out a syllable I want to manipulate, and I can send out an optogenetic trigger. And here we're going to use those optogenetic triggers to specifically cause dopamine release in dopaminergic axons that live within the dorsolateral striatum itself. So it's a very spatially localized manipulation of dopamine. And here we're going to do a kind of unusual thing. We're going to calibrate our optogenetic manipulation such that the amplitude of dopamine release that we observe out here roughly matches the amplitude of dopamine transients that we observe when animals are just spontaneously behaving. And practically what this means is that we're giving a single 200 millisecond long continuous wave pulse of light at extraordinarily low powers. And what that in turn means is that this kind of optogenetic dopaminergic manipulation we're making, which is spatially localized, is far more subtle in general than the sorts of dopaminergic manipulations people have made to implicate dopamine in behavior. And so it stood to reason, and we anticipated that this kind of manipulation should not change the animal's behavior at all. And indeed, if we measure kinematic parameters, either within a given syllable or throughout the experiment, we see that these kinds of calibrated manipulations in which we're manipulating dopamine at the same scale as dopamine is naturally fluctuating have essentially no consequence on the animal's ongoing movements. These dopamine transients that we're inducing don't initiate new movements, they don't change the kinematics of syllables, they don't change the animal's speed or force it to turn. In other words, these results suggest that these natural fluctuations in dopamine that accompany spontaneous behavior are almost certainly not causal, at least in dorsolateral striatum, for influencing the animal's ongoing kinematics. And we know this isn't just an artifact of our optogenetics not working, because if we just turn the light on now for 2 seconds instead of 200 milliseconds, now we see the usual and expected dramatic changes in the animal's movement in this other context. Okay? So it looks like these spontaneous fluctuations aren't changing the animal's ongoing kinematics. What about how often syllables are used? So here we see something very dramatic. So here we're just going to pick one syllable out of the maybe 8 or 9 that we've specifically targeted for optogenetic pairing and reinforcement. Here we're looking at some pre-stimulation data just to look at baseline expression of this particular syllable, which is the rearing syllable. I'm showing it to you here in green. And what we see is that nearly immediately, as soon as we turn on the light and begin to pair the expression of that rearing syllable with dopamine, within 2 or 3 pulses, the animal recognizes that there's some sort of relationship between this very subtle optogenetic manipulation we're making and the generation of that particular egocentric behavior. And it begins nearly immediately to upregulate the expression of that targeted behavior. It does it during the first session. We give it a little bit of a break. It does it again during a second section. And we know that this isn't just dopamine causing the upregulation of the targeted behavior, but instead it reflects learning. And we know that because the latency for the animal to express the targeted syllable on the second session is shorter than on the first session. And importantly, if we turn the light off, we see this kind of persistent expression of the targeted behavioral syllable, even in the absence of this optogenetic manipulation, consistent with the animal having learned an association between dopamine and this particular behavior, again, consistent with reinforcement. And I just want to mention what I think is kind of amazing here, which is that, of course, while we're doing this, the animal is just running around in a dark, empty bucket with no sensory cues from the outside world at all. So the only way the animal has to know that it's generating the targeted behavioral syllable is through efference copies and proprioception. But that is sufficient for the animal to understand its own behavior and to cause this kind of targeted upregulation. Okay, what about behavioral variability? We could look at trials in which we add a little dopamine and compare the animal's subsequent behavioral decisions to what we observed on those trials when we didn't add a little dopamine. And what we see is consistent with what I showed you in the endogenous data, which is that on those trials in which we add a little bit of extra dopamine to a given behavioral syllable, the very next behavior the animal generates tends to be a less likely or a more variable behavioral choice, whereas on those catch trials where we don't do that, the animal tends to do more predictable things. And so it's clear from what I've told you that these kind of spontaneous dopamine fluctuations don't seem to be influencing kinematics, but instead seem to be shaping the ongoing structure of behavior, both by influencing behavioral choices in the near future, and I think most importantly, by reinforcing over timescales of minutes the usage of associated behavioral syllables. And this suggests that maybe these spontaneous dopamine fluctuations that we're observing are acting as a kind of continuous teaching signal to sculpt the architecture of behavior. So we wanted to kind of query that formally. And so to do that, we've built a very simple reinforcement learning model in which we use actual data, actual dopamine syllable associations to train a reinforcement learning agent. And then we ask whether or not the behavioral choices that are made by this trained reinforcement learning agent match up with kind of held out observed behavioral choices that are made by real life mice. And if in fact they match up, it would suggest that in fact dopamine is acting as a kind of teaching signal. So here, just to quickly explain how the model works, the state that the model is in represents the syllable the animal is expressing at any moment in time. The action is the animal's choice of its next syllable. Here we, of course, substitute the traditional reward signal for the dopamine that's associated with any given syllable. And we use simple Q-learning rules to update a Q table, which here is equivalent to a transition matrix that governs how any syllable transitions into any other syllable. You'll notice here we're only going to focus on transitions amongst the top 10 syllables. That's because we're a little data limited. And you'll notice also that there's some syllable transitions we don't consider, and that's because these are syllable transitions that don't exist in the empirical data. That said, we initialize this transition matrix uniformly, and then we just kind of train a reinforcement learning agent using simple Q-learning rules, and we can learn a transition matrix that effectively reflects the behavioral choices this reinforcement learning agent would make if it were able to behave in the real world. And then we can compare this high-dimensional, this 100-dimensional transition matrix to an actual transition matrix from held-out data. And what's remarkable is that we capture a huge amount of the behavioral variation that's observed in real-life data when we just train an RL agent using these syllable-associated dopamine transients. And that really argues both that continuous variation in dopamine might be acting as a teaching signal, and that those signals might be interpreted by animals as reward-like signals. Just on this last data slide, I just want to mention that everything that I've shown you here is specific to dorsolateral striatum. If we go ahead and repeat the exact experiments I've been describing to you in dorsomedial striatum, we no longer see the correlations between dopamine and behavior that I described to you before. And if we try to optogenetically stimulate dorsomedial striatum, now we can no longer reinforce individual behavioral syllables. So to sum up, I've described to you at a kind of very high level motion sequencing, which allows us to atomize behavior into components, thereby revealing this kind of underlying structure to behavior. I've told you that it seems like dorsolateral striatum plays an important role in assembling the behavioral sequences that are captured by this behavioral state map, and that dopamine seems to play an essential role in affording this behavioral state map its structure. It does so not through controlling the animal's kinematics on a moment-to-moment basis, which is, I think, the kind of naive zeroth order prediction you would have made, but instead, in this kind of very surprising way, by acting as a teaching signal to shape the future usage and sequencing of behavioral elements during spontaneous behavior. And what's really gratifying about this is that, of course, this means that the role that dopamine is playing during spontaneous behavior is analogous to the kind of role that dopamine has been well characterized as playing during much more structured tasks when animals have rewards. It looks like the very same kind of circuits, and in fact, the very same computations that support kind of reward-based, goal-oriented learning seem to come into play during spontaneous behavior, suggesting a kind of unitary function for dopamine in controlling the overall architecture of behavior in multiple circumstances. And the key remaining question is, of course, why does dopamine vary? Right? What's controlling the variation in dopamine, thereby structuring behavior? That's a problem for another day. And so with that, I'll just mention that the work that I've showed you today was largely done by Jeff Markowitz, who just started his own lab at Georgia Tech, along with two great graduate students, Wynne Gillis and Maya Jay. And all this work is part of a broad collaboration I have with Bernardo Sabatini, Scott Linderman, and now with Cheeto. Thanks for your attention.
We have a few minutes for questions.
Of the syllable and text limits and also
Yes.
are these variables correlated with themselves, or they come from different parts of the variable?
That's a good question. So the reason why we built these encoding and decoding models was to get a sense of, there's going to be some correlation amongst all of them. The correlations are pretty weak. But we wanted to get a sense of, like, if we were to build an encoding model, for example, or a decoding model, what's the relative weight afforded to each one of these kind of behavioral features if you wanted to, for example, predict the fluctuations in dopamine. So it turns out that each one of these three things seems to play a different and complementary role in kind of recovering the dopamine transient. So all three seem to matter. And what's interesting is that if you look at the kind of the timescales at which the beta parameters evolve, it turns out that velocity is important at very, very short timescales, along with sequence variability, which is exactly what I showed you in the correlations. And usage tends to be important over much longer timescales, which is exactly what I showed you. Usage seems to be reinforced over minutes, whereas variability and velocity seem to be important over seconds. And actually, the models capture those temporal dynamics and demonstrate that it looks like, if you wanted to predict dopamine, you'd need all three of these different behavioral features to make accurate predictions.
Yeah, that was amazing how you pinned down these relationships between dopamine and the syllables. I'm wondering, though, is this kind of dopamine noise or reward noise that's happening in these situations where the animal's just walking around and you have these kind of micro, unmotivated or unexplained fluctuations that have no purpose, and the animal's behavior is modulated by it, but not in any purposeful or good way?
Yeah. So in my discussion, which there are many possibilities for where these dopamine fluctuations arise. And in fact, David Kleinfeld has recently published a paper in which essentially stochastic dopamine fluctuations in the cortex over much longer timescales than I'm showing you here. Right? These are sub-second fluctuations. His fluctuations are over many minutes. But high amplitude dopamine fluctuations in the cortex are seemingly appropriated, these random fluctuations are seemingly appropriated as kind of windows of learning that animals can take advantage of that shape behavior in this kind of accidental way. We think that this is not accidental, and we have ideas about where these fluctuations are coming from. And the reason we think it's not accidental is because a thing I kind of didn't have time to elaborate on, but which I think when you think about it is pretty obvious, is when you throw a mouse in a bucket, actually it has characteristic and stereotyped behavioral dynamics that evolve in a really reliable way across animals, across time. So when animals first get into the bucket, they're in this kind of exploration mode, then they kind of are in this like less exploratory mode, and then they're in this kind of chill out mode. And that's all very stereotyped. And so the fact that like behavior itself has very stereotyped temporal dynamics is difficult to reconcile between, and if dopamine is in fact reinforcing, it's difficult to reconcile those two things. And so we think it's not noise. I mean, formally it could be. It's choice number 3 in our discussion of possibilities, right? But I think it's likely to be something. We think it's likely to be the output of some sort of computation that's actually shaping the animal's behavior. And if you'd like me to speculate on what I think that computation is, I'd be glad to.
Yeah, I was intrigued by your observations of dopamine dips around the transition points of syllables. And it sort of aligned with a question that I generally have, which is that it seems like you're treating dopamine as a sort of prospective signal, and rather than looking at it as sort of a retrospective signal. And I'm wondering if you've looked at the explicitly the stoppage of syllables associated with the dip, since one syllable stops before another begins. And sort of related to that, if you, with your optogenetic stimulation, if you look at what's happening before your stimulation. I realize you're doing this in closed loop with a specific syllable, so it makes sense that that one is reinforced. But I guess I'm wondering about sort of your framework and thinking about it sort of prospectively rather than retrospectively.
Yeah, so you might imagine, right, that either just in the endogenous data when we look at correlations, or in the optogenetic data, that there might be some temporal slop, right? That like maybe when I reinforce a syllable, it doesn't just reinforce that syllable, but you might expect that it would reinforce like the behavior that led to that syllable, the syllable before, right? Retrospectively. Or you might expect that it could, maybe, be a teaching signal in the kind of anterograde direction. So we've done all of the sorts of things you'd want us to do, right? We've taken the dopamine signals and lagged them out forward and backwards to see whether or not the correlations still hold. In all of our models, we do that as routinely as a control. So it turns out that the reinforcement that we're predicting based upon an endogenous dopamine fluctuation really seems like it's temporally associated with the syllable that it's attached to. There is a little bit of slop, maybe, like the amount of reinforcement goes down by a factor of a log, okay, to the 2 temporally adjacent syllables, and that's it. We can do the same thing in the reinforcement learning model, right? I showed you this beautiful transition matrix. Like if we just shift over the relationship between behavior and dopamine by 1 syllable, that relationship goes to almost nothing. So it really is quite precise, actually, which is a little bit surprising and interesting. I'll say that we've built a model in which, like a prediction you might make is that those syllables that on average are associated with the most dopamine should be those syllables that show the greatest degree of reinforcement if our exogenous optogenetics is kind of being added to the endogenous fluctuations. That prediction turns out to be true. Again, kind of centering our attention on the syllable that we're actually targeting. But you're right, there is some slop in both directions. There's a little bit of retrospective slop, there's a little bit of prospective slop. I'll also mention, sorry, since I get to talk, right? I'll also mention one thing about this, which I think is really interesting. So, the duration distribution of the syllables has a mean that's like 400 or 500 milliseconds, but the actual implementation of syllables is often much longer than that. And what that means is that we're shutting the light off, and the dopamine transient that we induce is off nearly always before the syllable is completed. And yet, syllables stay intact. So if behavior were truly fully continuous and not discretized in any way, you might imagine that we would be reinforcing just the front half of each syllable, or just the part of the syllable that was associated with our optogenetic manipulation. But actually, that's not true. Syllable durations don't change at all when we do this optogenetic manipulation. Only usages change. And that suggests this kind of intriguing idea that maybe there's something discrete about behavior at this time scale, which is very consistent with the things that you were talking about earlier today, which I love that work about autocorrelations across cortex. And I very much have been thinking about the idea that maybe those kinds of timescales are setting these discrete timescales that are used by the brain to organize sensation and action into little bundles that make sense to the brain. That's a speculative idea we can talk about at dinner.
Great, fantastic. I'm sorry. Let's do it at poster time because of the poster session. So please hold your questions and bring them to Bob after.